Okay. Okay. Welcome, students. Welcome to the channel. Today, we are going to start with the new lecture series. As you know, we are here for the new lecture series on instrumentation in life sciences. So we'll be continuing with this series. Today is the intro lecture for this series. In this series, I am going to tell you about the instrumentation in life sciences, this add-on course, and we'll be covering few points in the syllabus that is being allotted to me. Okay. So I hope my voice is clear to all of you, and you are able to see what my screen looks like. If you're not able to see it clearly, just put the resolution to the highest in your YouTube so that it is visible to you properly. Okay, so as you know, this course is started in the Department of Biotechnology, Vivekanand College, Kolhapur. So we'll be continuing with this. First of all, let me give you an overall insight about this course, and then we are going to move further in this course. Okay. So if any problem, you can comment below in the section so that I'll be able to clear your doubts regarding that. Okay. So let's start with the lecture. I hope the sound and all is clear to you properly. Okay. So instrumentation in life sciences is a course which is started by the Department of Biotechnology a few years back. And the main objective of this course is to give the knowledge to the students about the different instrument that is used in life sciences. Life sciences contains lots of fields, for example, molecular biology, microbiology, biochemistry, yes, bioinformatics, genetic engineering. So lots of fields are there in biotechnology. So it contains lots of instrumentation that are required to do different kinds of experiments. Okay. So for that purpose, you require these instruments in every field of biotechnology or every field of biology in that sense. Okay. So this course provides you the details of each and every instrument that is to be used in life sciences. Okay. We combinedly call all these subjects in a common branch, that is life sciences. Okay. So we are going to see about the instruments in this course. Okay, so one by one I'll take you down to the different aspects of this course, that is syllabus, the syllabus that is distributed among the faculties of Department of Biotechnology, and we are going to see the distribution also at the practicals that will be taken by the different faculty in the Department of Biotechnology, Vivekanand College, Kolhapur. Okay, so myself, Mr. Ashutosh Upadhye, I am the coordinator of this course. So the introduction lecture for today will be taken by me. Along with the introduction, I'll be covering few points that is in the syllabus that is allotted to me. Okay. So first of all, let me tell you the teaching and non-teaching faculty that is present uh, in this course, that so that you will be clear that who all are going to teach you this course. Okay, so let me show you the slide for the teaching faculty here. So this following gives you the list of all the teaching faculties here. First of all, uh, myself acting as a coordinator plus faculty, teaching faculty. Then Mr. S.G. Kulkarni sir is there. He is the head of biotechnology entire department. Then S.H. Nadaf madam is there. She is the head of biotechnology optional department, Vivekanand College, Kolhapur. Then there are other faculties of the department, namely V.N. More, S.S. Patil, V.B. Kankekar, J.J. Gaundi, V.N. Arekar, R.M. Shetty, D.A. Vazantri, and S.D. Potdar. Okay, there are total 11 faculties that will be teaching you this course. The faculty code or the name faculty code for each one of them is given over here, so that you can see what all courses that are being allotted to these teachers. Okay, so let's see now the syllabus for the faculty that is distributed. You can see now the syllabus that is prescribed by the BOS Biotechnology for the instrumentation course in life sciences. Okay, so this instrumentation course in life sciences is given this syllabus which is present in front of you. You can see there are different eras or different instruments that are included which is used in life sciences. Okay, so first of all, the unit 1, it has general instrumentation, principal working and application of all the general instruments that are used that may be present in a biology lab. Okay. First of all, there will be digital weighing balance, pH meter, centrifuge, refrigerator, deep freeze, autopipetter, yes, water bath, dry bath, thermometer, mini spinner, then vortex mixer, magnetic stirrer. These are all the general instruments that are present in a general biological laboratory. Okay, then we are going to see instrumentation for specifically biochemistry and related subjects, its principle, working and application in details. Each lecture that will be taken by the faculty is of 1 hour, remember. Okay, so in biochemistry there are titer, then colorimeter, titrations are there, spectrophotometer are there. Okay, so I'm just naming out the instruments that will be used. Then BOD, COD apparatus along with its principle will be there. Then BOD incubator, specific gravity, then chromatography, different types of chromatography are there. TLC, that is thin layer chromatography, then paper chromatography, all the instrumentation that are required for that. Then ion exchange chromatography is there, gel filtration chromatography, dialysis, and refractometer. Okay, so these are the all the instrumentations that are required for biochemical analysis of water, and other samples, industrial samples, sewage water, etc. Then sugar sample is there, or purification of a protein is there. So in that case, we require such kind of samples. Okay. Then we are heading towards the instrumentation for microbiology. Okay. So in this, the instruments that are covered are autoclave, then digital autoclave, digital version of autoclave is also there, that will also be taught to you. Then hot air oven, then incubator, microscope of different types, may it be digital, confocal microscope, then light microscope, simple compound microscope. Okay, photographic microscope. Okay, then fermenter, shaker, incubator is there. Yes, distillation unit of different types, chemocytometer, replica unit and its technique. Okay, that also will be taught to you. Then other immunoassay techniques will be taught to you along with the colony counter. Okay, so these are the basic instrumentations in microbiology. Few more instruments are being added in the practical course that you are going to see. Okay, then the unit number 4 relates to the molecular biology. Here also we are going to see the principle involved, the working of the instrument along with its application, or what are the different processes that you can do with the molecular biology instrumentation. Okay, various kinds of gel electrophoresis machines we are going to see. Then we are going to see page gel, that is polyacrylamide gel electrophoresis. Today in this is the topic that I am going to teach you. So today in today's lecture, I'll be covering the gel electrophoresis and page. Okay, these two points I'll be covering in today's lecture. Then in this, electroblotting is there. Then UV transilluminator, PCR cooling centrifuge, liquid nitrogen homogenization, and liquid nitrogen crushing will be demonstrated to you, or and the theory behind it will be taught to you here. Okay. Then cryopreservation, how we can preserve a biological sample at minus 196 degrees in liquid nitrogen, that also will be taught to you in this molecular biology instrumentation part. Okay. And the last part is plant tissue culture. Principal working and applications of all the different techniques that are there, may it be asepsis, that is aseptic inoculation, or surface sterilization, instruments, that is hot plate, then laminar airflow, it vertical, horizontal, then rotatory shaker incubator, then cell counter. These all instruments will be taught to you in plant tissue culture and the plant tissue lab also. Okay. So this is the related syllabus that will be taught to you. Along with this, few advanced instrumentations will also be taught, such as spectrophotometer. Yes, then how to approach for TLC, HPTLC, HPLC, that also will be taught to you. Let me discuss the distribution of the syllabus. You can take a screenshot of this if you want. See, the distribution of syllabus is given here. The unit number 1 is consisting of 6 1-hour lectures, and this will be taught to you by JJ Gaundi madam and Poddar madam. Okay, so these two are going to share this unit among them. Then unit number 2, that is principles of biochemistry, will be shared by Mr. SG Kulkarni and SH Nadaf madam. Both of them are going to share the biochemistry part. Then the microbiological part, the biochemistry part is, by the way, having 10 lectures. Okay, the microbiological part is having 9 lectures and 3 teachers, More madam, Patil madam, and Vazantri madam. 3 teachers are going to take the microbiological part, and then the molecular biological part will be taken by myself, Mr. Ashutosh Lakshman Upadhye, and then VB Kankekar madam. Okay, the molecular biological part, 9 lectures are allotted to this, and that will be taken by us. Okay, then the last unit will be taken by Arekar madam and Shetty madam. The weightage given is 6 lectures. Okay, so this is the distribution of the syllabus. Then there are around 15 practicals that are included here. You can see there are 15 different practicals, and the faculty code is also given here. You can check out the code given at the starting of this video, where you know which teacher is going to take the which lecture. Okay, or which practical will be taken by which teacher. Okay, so this is all about the the syllabus and the practical theory syllabus that will be taught to you in this instrumentation course in life sciences. The scope of instrument is given, where the instrument will be used commercially. Means what? Suppose centrifuge is there, then it will be used in hospitals, biotechnological and pharmaceutical industries. Okay, then academic research institutes, hospitals, etc. Cell counting will be there in hospitals, research institutes, pharmaceutical industries. Okay, beverage companies, food and beverage companies. Okay, so which instrument is chromatography? All types of chromatography, all types of pharmacological companies requires chromatography for purification of substrates and all. Then academic research institutes, food industry companies, okay, environmental testing companies uses chromatographies. Okay, then electrophoresis, obviously all types of diagnostic labs, academic research, diagnostic centers, hospitals, pharmaceutical industries, and biotechnological companies. Then flow cyclometry, immunoassays for different types of diagnosis of different all kinds of diseases. You use immunoassay techniques. Okay, then microscopy, obviously it is present in each and every lab. Okay, so this is the instrument wise scope of the all the instrument that you are going to see in this course. So I hope you will be benefited a lot with this course. Now let me start with the point that I am going to teach you today. In the syllabus, if you see, I have already mentioned to you, I'm going to teach you with gel electrophoresis and PAGE. Okay, that is polyacrylamide gel electrophoresis. Okay, so let's start with the first point. First point, that is gel electrophoresis. Okay. Okay. So, agarose gel electrophoresis. Agarose gel electrophoresis is widely used technique to identify or to separate the DNA molecule. Okay. So, what is the importance of agarose gel electrophoresis that we are going to see? Okay. If the screen is not clear, students, adjust the resolution of your YouTube to maximum so that you can see it clearly. Okay. So now we are going to see agarose gel electrophoresis. It is a widely used technique for the analysis of nucleic acid as well as proteins. Okay, so in a molecular biology lab or any kind of diagnostic lab, you must have agarose gel electrophoresis. Okay, so earlier the electrophoresis used to be done on the paper or starch gel, etc., until the evolution of agarose or discovery of agarose. Okay, so it is a process that is used to separate different molecules. Okay, so what is agarose gel electrophoresis? It is a process that is used to separate different molecules. Okay, so you can see here. Okay. It separates molecules on the basis of their rate of movement through the gel under the influence of electric field. Okay. So what happens in agarose gel electrophoresis? You apply an electric field on the sides of the chamber, electrophoretic chamber, which is generally present. And in that chamber, you use a solid matrix to separate the biological molecule. Okay. So if you are wanting to separate a biological molecule, you have to either use its molecular weight or you have to use its electric charge, okay, to separate the molecule. Okay, so here we will be talking about the agarose gel electrophoresis to determine different kinds of products. Okay, it can be DNA, PCR of DNA. Okay, then presence of different kinds of DNA sample, protein samples, etc. We are going to see. Okay, this kind of tube is there that is called as Eppendorf tube or vial. Okay, this is a vial. Okay, that is generally made of plastic. Okay, it is made up of autoclavable plastic. Okay, that can be autoclaved so that you can store the DNA. Okay, you know DNA is having a property. Okay, DNA is having a property in which DNA binds to the glass. Okay, DNA has a property. DNA, if you take the DNA sample in glass, it binds to it. Okay, so it is not easily separatable. Okay, so therefore we use this kind of plastic tubes that is called as vials. Okay, so DNA is negatively charged. Okay, so generally more than proteins, agarose gel electrophoresis is used to separate DNA. Remember, okay. So DNA is a negatively charged molecule. You know it, okay. So since it is a negatively charged molecule, it can be separated in the electric field easily because it is going to migrate towards the positive charge. So you can see here DNA is migrating towards the positive charge from this side to this side. Okay, so this is a gel made up of a polymer called as agarose. Okay, we are going to see how the agarose forms a gel in details. Then it is a polymer that is agarose that helps to form a gel. Okay, and there is a slow movement of the DNA bands across this. Okay, now the agarose here, the concentration of agarose is different. Okay, now what is agarose? You know, agarose is isolated from a algae. Okay, agarose is isolated from a seaweed or sea algae. Okay, this the name of this algae is Gelidium corneum. Okay, this is an algae that is red algae. It is a type of red algae. Okay, that is present in seabed, and from this the agarose is isolated. Remember this. Okay, so what is agarose? Agarose is a disaccharide. Okay, agarose is a disaccharide polymer. Okay, polymer made up of a disaccharide. What is agarose? Agarose is a disaccharide polymer, means a polymer made up of disaccharide that is composed of two different sugars. Okay, two different sugar. One is galactose, one is galactose. Okay, one is galactose plus 3,6 N hydro galactose. Okay, so what is agarose? Agarose is a polymeric disaccharide called as agarobioes. Okay, it is a polymeric disaccharide called as agarobioes. Okay, so agarobioes is a disaccharide made up of galactose plus 3,6 N hydrogalactose. Okay, now this agarobioes is repeated again and again to form a polymer of agarose. Remember, So agarose is generally a bit expensive since it is isolated from a C algae. Okay, so you can see a scanning electron microscope image of the agarose gel. Okay, the agarose gel piece is taken and an electron microscope image is taken here. You can see how it looks under microscope. Okay, so this is the image of polymerized gel. Okay, so how fast the DNA migrates depends on what? It depends on the strength of electric field. Okay, whatever the electric field that you are applying, that strength is responsible for movement. Okay, so the more the agarose you put, smaller will be the pore size. Okay, more the agarose you put, or the concentration of, if the concentration of agarose increases, the pore size decreases. Okay, okay, so you will be able to you will not be able to separate larger molecules if the pore size is less. Okay, so depends on agarose concentration. Typically for general nucleic acid we use 1% concentration of agarose, okay, in water. Okay, so how do we prepare the agarose gel? Okay, so I was talking about the different factors. First is agarose concentration. So it you know, is responsible for mobility. Okay, mobility of the DNA molecule. First factor, that is concentration of agarose, is very important for the mobility of the DNA molecule in the gel. Second is electrical field. Okay, electrical field. If you provide larger electric field, then what is going to happen? The molecule is going to move faster. Okay, but 50, maximum 50 to 100 volt field is applied. If you apply more field, then gel is going to melt. Okay, then it is also responsible, the movement of the DNA or the biomolecule in the agarose, generally DNA, also depends on the molecular weight. Okay, if the molecular weight of the DNA is less, then it will move faster, and if the molecular weight is more, it will move slower on the agarose gel. Okay, so these are the three factors on which the molecular weight depends. It is properly told to you, smaller molecule migrates faster and the larger molecule will migrate slower. Okay, okay, is it clear to all of you? Okay, so this is how the agarose looks like. You can see it's like a powder. This is the structure of the agarose that is shown to you here. I'll zoom at it. This is the structure: galactose and 3,6 anhydrogalactose. It is a agarose, that is a disaccharide. Okay, so this disaccharide is repeated again and again in this powder, and when you boil it, it is going to form a gel. Okay, so it was first time used by Robert Koch to isolate the bacterium for tuberculosis. Okay, so the tuberculosis bacterium is Mycobacterium tuberculosis. Okay, this bacterium was first time isolated on an agarose plate made up of agarose polymer on that. Okay, so how to make a gel? See, first of all, you have to use a buffer. Generally the buffer is different kinds of buffer. You use it for preparing agarose. In water also you can prepare agarose, but the thing is it should have a pH of 7. Okay, so in order to stabilize. Okay, so generally you can make general buffer that is used is TAE buffer. Okay, TAE buffer in 1x concentration is used to prepare agarose gel. TAE is tris acetate EDTA. Okay, TAE is tris acetate EDTA buffer. Okay, so this buffer is very important because agarose gel is prepared in this buffer. Okay, suppose if I want to prepare 1% agarose gel, what I will be going to do? I will take 1 gram agarose powder plus 100 ml of water. Okay, distilled water or TAE buffer. Okay, or TAE buffer, 1x TAE buffer. Okay, generally we prefer TAE buffer because it has better pH balance. Okay, then we are going to boil it. Okay, okay, until forms a clear liquid. Okay, clear liquid. Okay, so once it forms a clear liquid, then what we are going to do? We are going to cast the gel. Okay, we are going to cast the gel in a casting tray. I'll show you the diagram of casting tray also. Okay, you are going to see this in your practicals also. So don't worry. Okay. So this is the electrophoretic equipment. This is a gel tank which is used to do the electrophoresis. It has two different, uh, units. This is the positive and negative terminal. Then this is a power pack. Okay. Then lid cover. Then there are combs. Then this is a casting tray. Okay. Here the comb is attached like this. Okay. I'll show you. See, this is the tray, and these combs are attached this way, okay, like this, and here also like this. Okay, at the desired length it can be attached. Okay. Okay, at the desired length these combs can be attached, and when these combs are attached, then what you can do. Okay, so you can cast the gel. Okay, the tray has to be packed from both the sides. You see here, we have applied a tape over here so that the gel is trapped inside this. And when the gel is trapped, you pour the agarose here, and it will align, and these kind of wells will be formed. These wells, in these wells, the DNA sample will be added. Okay, so these wells are quite easily seen in the diagram which was which we saw earlier. See here, these are the wells. These are the wells. These wells are created by that comb. Okay, that comb size also matters, and the size of the gel depends on that comb. Okay, so this is the casting tray. Then after putting agarose, you have to add a buffer solution, generally DAE buffer solution. Then after putting agarose, it looks like this. Okay, but when you boil it, it looks like this. It forms a clear liquid. Okay, so after boiling, you have to carefully pour the gel onto the gel. Uh, After slightly cooling at 60 degrees Celsius. Okay, at this point you have to add a visualizer, that is ETBR. Okay, so ETBR, generally DNA, we cannot see the DNA, remember. Okay, so we have to add ETBR for its visualization. It is a visualizing dye. It is a visualizing dye. Okay, so this dye is very important. This dye is very important. Why it is important? See, it is a visualization dye. Okay, okay, it's a visualization dye. Okay, so it is made up of ethidium bromide. Okay, so what does this dye do? This dye enters into the bases of the DNA or the ribose sugar of the DNA and interchelates between them. Okay, when it interchelates, it forms interchelates, and when this DNA gel is exposed to the UV, then what happens? It visualizes and shows orange color fluorescence. Okay, so this is what, see, these are the wells here. You can see the different kinds of wells here. These are the well number one, lane number two, lane number three, lane number four. Okay, and the DNA is electrophoresed by connecting it to power plug. Okay, power plug, and the DNA moves in the positive side. Okay, this is the positive side. Okay, and this is the negative side. So the DNA moves towards anode. Okay, DNA being negatively charged move towards the anode. Okay, so when the DNA moves here, what will happen? Gradually there is a separating of DNA bands. Now, regularly the DNA solution that is taken in vials is not visible. Okay. Okay, DNA solution is not visible. Okay, so when we are loading the DNA onto the agarose gel, so at that time we have to add DNA solution along with the dye. Okay, DNA solution has to be added with the dye. Okay, this dye is generally called as gel loading dye. Okay, gel loading dye. Okay, this is made up of different components. First is bromophenol blue. This is a dye. Okay, then xylene cyanol. Okay, then it is having glycerol or sucrose. Okay, this glycerol or sucrose is for giving weight to the DNA. okay, giving weight to the DNA. These are the tracking dyes. This keeps a track of the DNA while moving in the electrophoresis. Okay. Then there is pH stabilization buffer. Okay. So all these are the all different components of the gel loading dye that is to be added into the gel. Okay. See here, this looks like this blue color. Okay, this looks like a blue color. Okay, blue colored dye, so that we can see the sample. Okay, generally this dye is available in 6x or 3x or 2.5x concentration. We have to make it 1x and then we have to use it for electrophoresis. Okay, then using a micropipette, this is the micropipette that you can see here. Using a micropipette, that wells that we have created has to be carefully loaded with the DNA. Okay, so using a micropipette that is finely tuned and smaller quantity of DNA can also be loaded onto the agarose gel, that is put or the loaded into the DNA well that we have prepared just earlier. I have shown the diagram to you. In that well we have to load the DNA. We have to carefully do it so that the well is not punctured. Okay, if the well is punctured, then the DNA sample will be all over the gel. Okay, it has to be moved properly in a sequential way. So we have loaded the sample properly. Okay, then electrophoresis, we have to start the electrophoresis. You can see here both the positive and negative terminal is attached to the power pack, and the electrophoresis is started. This is the DNA sample. Here you can see this is the DNA sample. I hope it is visible to you. Let me make it darker. This is the DNA sample that will move forward like this. Okay, okay. Then this the tracking dye separates here. The dye which is moving forward is bromophenol blue, and the dye which remains backward is xylene cyanol. Okay. Uh, okay. So this is how the separation of the DNA takes place on the basis of size. 12,000 base pair DNA will remain behind due to its higher molecular weight, and the smaller DNA will move forward. This is a DNA marker lane. Okay, these are the different bands of DNA marker. Okay, which separates depending on the size. Okay, and this is the sample DNA. These are the sample DNA. Okay, this is the sample DNA, that is blue colored sample DNA, that is sorry, this is a dye, and DNA is migrating here. Okay, now while migration you cannot see the DNA. Okay, so after the electrophoresis is done, you have to check the DNA onto the UV transilluminator. Okay, so I told you this ETBR, we have added this ETBR at the starting when while preparing the gel. If you don't add the ETBR at the starting, you can add it at the last after staining. Means what? After electrophoresis, you have to stain the gel again then. Okay. But if you are adding it at the starting only, you can easily visualize the gel directly. Okay. So this ETBR is a powerful mutagen. Remember. So while using ETBR, you have to be very careful. It is a very powerful mutagen, and it is moderately toxic. So always gloves, you have to use gloves at all times while using ETBR. Okay, so it is a toxic chemical, and this uh, carcinogenic also. Okay, some safer alternatives. So while it is toxic, why you cannot use other stains? You can use different stains, other stains that are available for different companies, that is mentioned over here. So what is the advantage? They are inexpensive, less toxic, no UV required, then directly you can see the DNA. Okay, disadvantage is sensitivity is less, more DNA is needed onto the gel. If the DNA is very less, then it is not able to show you the DNA. Okay, so in that case ETBR is only used. Okay, so you can see after the removing the gel after electrophoresis, you have to put the gel into the staining solution. You have to add ATBR, and then you have to visualize the DNA into the UV chamber. Okay, so the blue colored, this is actually a box you can see, which shows up the UV light. Okay, you have to put the DNA in the UV light, and you can visualize the DNA, and the DNA looks like a orange band. Here you can see the orange band is shown to you here. Okay, so DNA is a orange colored band that is visible in the agarose gel. Okay, so this is a black and white image of the DNA where you can see the DNA. You can see after the electrophoresis, the DNA looks like this. Okay, so this was all about the agarose gel electrophoresis. I hope you have understood how this electrophoresis is done. Okay, so while we will be doing the demonstration of the practicals to you, you will able to see the whole procedure again in the practicals also. Okay, so I hope you are getting the whole video here. Is it clear to all of you? Please, if you have any doubt, reply in comments. Is it okay? Okay. So, likewise, we are going to see other topics in the unit number 4, that is molecular biology instrumentation. So, let's start with the SDS page. We'll start with the SDS page tomorrow. Thank you for now, all of you. All the lectures from this lecture series will be taken like this only. If any doubt, you can ask in comment section below. The teachers will be always there for you to answer your questions. Okay. Thank you so much for students. Thank you so much for joining, all of you.